Hi everyone and welcome to Almighty Choice Tutorials. I'm really excited to have you here for our first VCE Biology Unit 2, Lesson 1. If you are a Year 11 biology student in Victoria and this course has been designed specifically around the current VCAA biology study design, my aim is not just to help you pass your SEC and exams but to help you genuinely understand biology. This week, we are beginning with one of the most important topics in genetics, genes, chromosomes, and genomes. By the end of today's lesson, you'll be able to understand what DNA is, how it is organized inside the cells, why humans inherit characteristics from their parents, and how scientists study chromosomes using karyotypes. Everything builds from today's lesson. So if you understand this chapter well, the rest of the unit 2 will become much easier. Before we begin, if you are new to the channel, don't forget to subscribe and turn on notifications so that you never miss a VCE biology lesson. Alright, let's get started. At the end of this lesson, I would like you to pause and ask yourself these questions. Can I define a genome, a gene, and an allele without looking at my notes? Can I explain what homologous chromosomes are? Can I distinguish between autosomes and sex chromosomes? Can I interpret a simple human karyotype? And finally, could I confidently answer a VCAA-style biology question on today's topic? If the answer is yes to all of those, then you've achieved today's learning goals. If not, don't worry, that's exactly why we have the workbook and homework booklet to reinforce what you have learned. Before learning something new, it's always helpful to activate what you already know. Take a moment to answer these questions. You can pause the video if you would like. Don't worry if you're unsure about some of the answers. This isn't a test and simply a way to connect today's lesson with the concepts you've already learned in Year 10 Science. Go ahead, have a go, pause for about 15 to 20 seconds. Let's walk through these questions together. Questions are: ask which organelle contains the cell genetic material in eukaryotic cells. The answer is nucleus. Remember that the eukaryotic cells have a membrane-bound nucleus, while prokaryotic cells don't. Question 2 says, what tightly coiled DNA is called? The answer is chromosome. When DNA isn't being used, it coils tightly so it can fit inside the nucleus. Question 3 says, which type of cell division produces identical body cells? That's mitosis. Mitosis is responsible for growth, repair, and replacing damaged cells. Question 4 asks, which cells are produced by meiosis? The answer is gametes. Sperm cells in males and egg cells or ova in females. These ideas are very important because today's lesson builds directly on them. If some of those questions were difficult, don't worry. By the end of today's lesson, they'll make much more sense. Let's check how you went. Now that we've refreshed our prior knowledge, let's begin exploring DNA. DNA is one of the most fascinating molecules in biology. Every single living organism on the earth has DNA. Whether we are talking about bacteria, plants, or animals, or human, DNA stores the information needed for life. But have you ever wondered why children usually look like their parents? Or why brothers and sisters look similar but aren't exactly the same? The answer lies inside DNA. Everything we are going to study over the next few weeks begin with the understanding of this incredible molecule, DNA. So, what exactly is DNA? DNA stands for deoxyribonucleic acid. That's a long scientific name. But you don't need to memorize every letter today. The important thing is understanding what DNA does. Think of a DNA as the body's instruction manual. Imagine buying a new piece of IKEA furniture. It comes with an instruction booklet that tells you exactly how to build it. DNA works in a similar way. Instead of building furniture, DNA provides instructions for making proteins. Those proteins control almost everything that happens inside your cells. They determine how your body grows, repairs itself, and functions every day. DNA also carries a hereditary information that you inherit from your parents. That's why families often share similar characteristics such as eye color, hair texture, or blood group. Here's an amazing fact. If you stretched out all the DNA just from one human body cell, it would measure around 2 meters in length. Yet, somehow, all of that DNA fits inside the tiny molecule that's called nucleus. Later in today's lesson, you'll discover exactly how that's possible. Many students believe DNA is only found inside the nucleus. That's a common misconception. The nucleus contains most of the DNA in eukaryotic cells, but it's not the only place where DNA is found. Let's compare different types of cells. In eukaryotic cells, which include animal, plants, and fungi, most DNA is stored inside the nucleus. However, two other organelles also contain DNA. The first is the mitochondrion. The mitochondria are often called powerhouse of the cells because they produce energy. Interestingly, they contain their own small circular DNA. Plant cells have another organelle called chloroplast. Chloroplasts also contain their own DNA because they carry out photosynthesis. Now let's compare that with prokaryotic cells, such as bacteria. Prokaryotes don't have a nucleus. Instead, their DNA is found in a region called nucleoid, which is located in the cytoplasm. That's a very common VCAA exam question. If you are asked where DNA is found, remember this. Most DNA is in the nucleus. Some DNA is in the mitochondria. Plant cells also have DNA in chloroplasts. Prokaryotes have DNA, but they don't have a nucleus. Take a moment to compare the diagram on the slide. Notice the difference between these cells. Let's spend another minute looking carefully at the diagrams. On the left, you can see a eukaryotic cell. Notice the large nucleus in the center. That's where the majority of the DNA is stored. You can also see the mitochondria. Remember, they contain a small amount of DNA. If this were a plant cell, we would also include chloroplasts. which contains DNA as well. Now look at the bacterial cell. Can you see what's missing? There's no nucleus. Instead of the DNA sits freely in the nucleoid region. This difference between eukaryotic and prokaryotic cells is one of the easiest way to distinguish them in biology. Whenever you see a VCAA question comparing these cell types, start by asking yourself, does the cell have a nucleus? If yes, it is a eukaryote. If not, it's a prokaryote. That's a simple trick you can help you pick up easy marks. Let's check your understanding with a quick true or false question. Take a few seconds to think about your answer. Prokaryotic cells contain DNA in a nucleus. Do you think that's a true or do you think it's false? Pause the video if you would like a little more time. The correct answer is false. Remember, eukaryotic cells do not have a nucleus. Instead, their DNA is found in the nucleoid region of the cytoplasm. This is one of the biggest difference between prokaryotic and eukaryotic cells. A simple way to remember it is eukaryote enclosed, prokaryote no nucleus. That's an easy mark in the exam if you remember the difference. Now let's have a look why this answer is false. Although bacteria and other prokaryotes definitely contain DNA, they don't have membrane-bound organelles. That means they don't have a nucleus. Instead, their DNA forms a circular chromosome located in the nucleoid region. Some bacteria also contain tiny rings of DNA called plasmids. We will look at the plasmid in more detail later in the course. For now, remember this important comparison. Eukaryotic DNA is mainly found inside the nucleus. Prokaryotic DNA is found freely within the cytoplasm in the nucleoid region. If you can clearly explain the difference, you will be well prepared for VCAA question. Now we know where DNA is found. Let's explore what DNA is actually made of. DNA is built from repeating units called nucleotides. Think of nucleotide as a building block. Just like a brick helps building a house, thousands and thousands of nucleotides join together to build one DNA molecule. Every nucleotide has three parts. The first part is a phosphate group. The second part is deoxyribose sugar. And the third part is nitrogen base. These are four different nitrogen bases: adenine, thymine, cytosine, and guanine. These four bases are incredibly important. You will see them throughout the VCE Biology. So it's worth becoming familiar with their names now. Don't worry if you can't memorize them immediately. By the end of this course, you will know them really well. Let's look more closely at how these nucleotides join together. The phosphate group and the deoxyribose sugar connect to form what we call sugar-phosphate backbone. You can think of this as two sides of the DNA ladder. The nitrogen bases stick out from the backbone and point toward the center. Later, these bases pair together to form the rungs of the ladder. Notice the diagram carefully. The sugar and the phosphate repeat over and over again all the way along the DNA molecule. This repeating pattern gives DNA both flexibility and strength. Whenever you see a diagram of DNA in exam, try identifying the sugar-phosphate backbone first. Then identify the nitrogen bases. This strategy makes interpreting DNA diagram much easier. Now we come to one of the most important ideas in genetics. The nitrogen bases don't pair randomly. They always follow strict pairing rules. Adenine always pairs with thymine. Cytosine always pairs with guanine. Never any other combination. Many students remember this with the simple phrase A with T and C with G. Let's practice those rules. The DNA strand shown on the screen reads A, T, C, G, A. Your task is to write complementary strand. Pause the video and try it yourself before looking at the answer. Remember, you don't rush, work through one of the base at a time. This is exactly the kind of skill that appears regularly in VCAA biology assessment. Now, let's see if you got your answer correct. So A will pair with T, T would pair with A, G and C, C and G, and A and T. Let's try another one and work through the answers together. The first base is A. It pairs with T. Next comes T that pairs with A. Then G pairs with C. C pairs with G. The second C also pairs with G. The next base is A, so we write T. Finally, T pairs with A. So the complete complementary strand is T, A, C, G, G, T, A. Notice that we did not memorize the whole answer. We simply applied the base pairing rules one base at a time. That's exactly how you should approach these questions in your SAC. Now guess, always use pairing rules. Excellent work. If your answer matches the one on the screen, you've understood complementary base pairing. If you made a mistake, don't worry. Most errors happen because students accidentally pair adenine with cytosine or forgot one of the rules. Keep repeating this simple pattern. A with T, C with G. You will use this knowledge again and again whenever we study DNA replication, mutations, and protein synthesis later in the course. So, it's well worth mastering now. Before we move on, here is something to think about. We've learned what DNA is and how it's built. But here is the next question. If every human cell contains about 2 meters of DNA, how does all of that DNA fit inside the tiny nucleus? That's exactly what we'll discover in the next section when we explore gene, chromosome, and genomes. Before we move on, let's check that you've understood the structure of the nucleotide. Read the 4 options on the screen. Which one correctly describes a nucleotide? Take a few moments to think about it before I reveal the answer. The correct answer is option C. A nucleotide contains 3 components: a phosphate group, deoxyribose sugar, and a nitrogen base. Many students accidentally choose an option with only 2 components or confuse a nucleotide with a nitrogen base. Remember, the nitrogen base is only one part of the nucleotide. The entire nucleotide always contains all 3 parts. Let's explain why. Option C is the correct answer. Every nucleotide follows the same basic structure. The phosphate group and sugar join together to form the outside of the DNA molecule. The nitrogen base points toward the center where it pairs with another nitrogen base. Whenever you're answering VCAA multiple choice questions, don't simply memorize the answer. Ask yourself, can I explain why the other three options are incorrect? That's a great way to strengthen your understanding. Let's pause and review what we've learned so far. Question 1 asks, what does A in DNA stand for? The answer is acid. The question 2 asks, name an organelle other than the nucleus that contains its own DNA. The answer is mitochondria. If you're thinking about plant cell, chloroplast would also be the correct answer. Question 3 asks for a complementary strand for A, T, G. Let's walk through it together. A pairs with T, T pairs with G, and G pairs with C. So the complementary strand is TAC. Finally, what is the overall shape of DNA? The answer is double helix. If you've answered these questions correctly, you are building a really strong foundation. So now we are moving into one of the most important sections of today's lesson. Students often confuse the terms DNA, genes, and chromosomes and genomes. The good news is that once you have understood how they are connected, biology becomes much easier. Instead of thinking of them as separate ideas, think of them as different levels of organization. Let's build that picture together. Earlier, I mentioned that every human body cell contains around 2 meters of DNA. Now imagine try to fit 2 meters of string into something smaller than the tip of a pen. That sounds impossible. So how does the cell manage it? The answer is organization. DNA wraps around proteins called histones, forming structure that becomes more tightly packed until they form chromosomes. This incredible packaging system protects the DNA and makes it easier for cell to divide. Without this organization, DNA would become tangled and damaged. Nature has developed an amazingly efficient way of storing genetic information. This slide shows one of the most important diagrams you will see in Unit 2. Let's read it from the largest level down to the smallest. At the top is genome. The genome is a complete set of DNA found in an organism. Within the genome are the chromosomes. Each chromosome is one long DNA molecule that has been tightly coiled. If you zoom into chromosome, we will find DNA. Along the DNA molecule are thousands of genes. Each gene contains instructions for making a specific protein or influencing a particular trait. Finally, genes can exist in different versions called alleles. For example, you might inherit one allele from brown eye and another allele for blue eye. The order is extremely important. Let's say together: genome, chromosome, DNA, gene, allele. I would like you to pause the video and repeat that sequence aloud 3 times. It will help you to remember it much easier. Now we are going to connect everything we have learned so far. One of the biggest challenges for the VCE biology students is remembering the difference between a genome, chromosome, DNA, gene, and an allele. In fact, they all are connected. Think of them as different levels of organization. Let's work through them one at a time. We'll start with the biggest level and work our way down. First, we have a genome. A genome is all the genetic material in an organism. In other words, if you collect every chromosome from one human cell together, they would make up one human genome. You can think of the genome as the body's complete instruction manual. Everything needed to build and maintain our body is stored within the genome. Now let's zoom in a little closer. The genome is made up of chromosomes. Humans have 46 chromosomes arranged in 26 pairs. Each chromosome is one very long DNA molecule that has been tightly coiled so it can fit inside the nucleus. Remember what we have learned earlier. If we stretch the DNA from just one body cell, it would be about 2 meters long. Wrapping it into the chromosome allows it to fit neatly inside the tiny cell's nucleus. Now let's zoom it even further. Each chromosome is made of a DNA molecule that carries a genetic instruction for the life. It tells our cell how to grow, develop, repair themselves, and produce protein. So the chromosomes are made up of DNA, but DNA itself contains smaller sections. Those smaller sections are called genes. A gene is simply a section of DNA that contains instructions for making one specific protein or influencing one particular trait. For example, there are genes involved in eye color, blood group, hair, texture, or thousands of other traits. Now, here is the final step. Although everyone has the same gene, we don't have the same alleles. An allele is simply a different version of the same gene. Let's look at the example on the screen. Here we have the eye color gene. Different people may inherit different alleles of this gene. One person may inherit the allele for brown eye, another inherit the allele for blue eye. Someone else may inherit allele for green eye. Notice something very important. Gene is the same. Only the version of the gene or allele is different. That's exactly what creates variation between the individuals. Now, I would like you to remember this sequence. Let's read it together. Genome, chromosome, DNA, gene, allele. Let's say it one more time. Genome, chromosome, DNA, gene, allele. If you can remember that, you will find many VCE biology questions much easier. Now, let's look at one of the ACT exam tips. Genes occupy a specific position in a chromosome, and that position is called locus. Every homologous chromosome has the same gene at the same loci. The alleles at those loci may be different. We will see exactly how that works when we learn about the homologous chromosome in the next section. Let's now focus on human chromosomes. Every normal human body cell contains 46 chromosomes. These chromosomes are organized into 23 pairs. 22 of these pairs are called autosomes. They contain genes responsible for most of our body traits. The final pair are sex chromosomes. Females usually have XX chromosome. Males usually have XY chromosomes. Notice something very important. Every chromosome pair contains one chromosome inherited from mother and one inherited from father. That's why we inherit traits from both parents. Now, here's an interesting question. If our body cells contain 46 chromosomes, why do sperms and egg cells contain only 23? That is a question we will answer properly in our next lesson, meiosis. For now, it's enough to remember body cells, also called somatic cells, are diploid, meaning they contain 46 chromosomes. Gamete, sperms, and egg cells are haploid, meaning they contain 23 chromosomes. When fertilization occurs, Sperm contribute 23 chromosomes and the eggs contribute another 23 chromosomes. Together, they restore the total of 46 chromosomes in the new individual. That's why chromosome number stays constant from one generation to another. Now, let's talk about homologous chromosome. That's one of the concepts that students often find confusing at first, but it's quite logical. Humans have 23 pairs of chromosomes. Each pair is called a pair of homologous chromosomes. One chromosome comes from the mother and the matching chromosome comes from your father. These chromosomes are same size and shape and they carry the same gene in the same position known as loci. However, they don't always carry the same alleles. For example, both chromosomes may contain the gene for eye color, but one chromosome might carry the allele for brown eye, while other carries the allele for blue eye. That's the one reason why offsprings inherit a unique combination of characteristics. Now, let's see whether you can apply what you have learned. Question 1 asks, which structure is a section of DNA that codes for a protein? Think carefully. Is it DNA or is it a chromosome or is it something smaller? You can pause the video and try answering all the 4 questions before we look into the answers. Let's check your answers. A section of DNA that codes for the protein is called a gene. Many genes together are found on a chromosome. All the DNA in an organism is called the genome. Now let's have a look at this sequence. Many students get this wrong because they mix up the chromosome and DNA. Here is the correct order from the smallest to the largest. A gene is a part of a DNA molecule. DNA is tightly packed into chromosome. All the chromosomes make up genome. So the one way to remember it is to imagine a library. A gene is like one recipe. DNA is like the page that contains many recipes. A chromosome is like a recipe book. A genome is an entire library. Visualizing it may help you to understand the hierarchy in a much easier way. Here's another quick check. Genes are larger than chromosomes. Is it true or false? Take 5 seconds to decide before we look into the answer. The answer is false because the genes are a smaller section of DNA that are located on chromosome. Let's explain this little further. Imagine unwrapping one of the chromosomes. Inside that chromosome is one incredible DNA molecule. Along that DNA molecule are thousands of individual genes. So a chromosome is not a gene. It is actually made up of genes. That's why chromosomes can influence many different traits. Whenever you say the word gene and chromosome in an exam, ask yourself, am I talking about one instruction or many instructions? One instruction is gene and many genes together are organized into a chromosome. Now, let's look at something that surprises many students. Different organisms have completely different chromosome numbers. Humans have 46 chromosomes, fruit flies have 8, rats have 42, bananas have 22, and potatoes actually have 48 chromosomes. Here's an important question. Does having more chromosomes mean an organism is more complex? Many students immediately say yes, but the answer is actually no. Chromosome number has actually nothing to do with the intelligence or complexity. It is simply characteristics of one species. That is a very common misconception. So make sure you remember it. Let's move on to one of the most useful tools in genetics, the karyotype. A karyotype is an organized picture of all the chromosomes in a cell. Scientists arrange chromosomes into the matching pairs, starting with the largest pair and ending with the smallest pair. This organization makes it much easier to identify abnormalities and compare chromosomes. Karyotypes are used in hospitals, research laboratories, and fertility clinics all over the world. They are an excellent example of how biology directly helps people in medicine. Let's compare the karyotypes of several different organisms. Take a moment to observe the images carefully. Notice that every organism has a different chromosome number. Ask yourself, which organism has the most chromosomes? Which has the fewest? Do you think organism with the most chromosome is automatically the most advanced? The answer is no. As we've already discussed, chromosome number does not indicate biological complexity. Instead, chromosome numbers simply reflect the evolutionary history of that species. That's why scientists don't judge complexity by counting the chromosomes. Let's discuss in more detail that the chromosome number does not determine the complexity of an organism. For example, potatoes have more chromosomes than humans. That certainly does not mean that potatoes are more complex. Chromosome number varies naturally between species. What's important isn't the number of chromosomes, it is the information contained within the DNA. That's another idea that VCAA examiners like to test because many students assume more chromosome equals greater complexity. Now you know that's not true. You've learned about chromosome number, and karyotypes. Next, we will explore another big idea in genetics, alleles. We will discover why humans are 99.9% genetically identical, yet every person is unique, and how different versions of genes create a variation we see around us. Let's see how well you have understood today's lesson so far. Look at the questions on the screen and pause the video for a while to answer these questions before I show you the answers. Now let's quickly have a look at the answers. A gene is a section of a DNA. Many genes are found in chromosomes. All the chromosomes together make up the genome. And in a karyotype, the chromosomes are organized into the matching homologous pairs from one largest pair down to the smallest. If you can explain that sequence confidently, you are well on your way to mastering today's topic. We are going to discuss about alleles in more details now. Here is something really fascinating. Did you know that every human being on earth is about 99.9% genetically identical? That means all the differences we see, eye color, hair color, height, blood group, and thousands of other characteristics come from only about 0.1% of DNA. So where do these differences come from? They come from alleles. An allele is simply a different version of the same gene. Think of it in this way. Imagine every student in your class owns same biology textbook. The textbook is a gene, but some students have the first edition, while some others have the revised edition. Those different editions are like alleles. The information is mostly the same, but there are some differences. Let's explore this idea further. Although people have same genes, they don't always have the same alleles. For example, everyone has genes that influence eye color, but one person may inherit an allele for brown eye, while another inherits an allele for blue eye. That's why people look different even though they share the same genes. Every body cell contains two copies of each gene. One comes from mother and another comes from father. So every person has two alleles for most genes. Those two alleles together influence the traits which is expressed. This idea becomes incredibly important when you study inheritance over the next few weeks. Let's look at a simple example. The gene for earlobe attachment has different alleles. One allele produces free earlobe, another produces attached earlobes. Notice something important. The gene hasn't changed. Only the version of genes is different. That's exactly what an allele is. Different version, same gene. Whenever you hear the word allele, immediately think different versions of the same gene. Alleles don't just affect earlobes, they are responsible for variation in thousands of traits. For example, blood group, flower color, seed size, plant height, hair color, and many more. Humans have over 20,000 genes. Most of these genes have different alleles. That's why every person is genetically unique. At the bottom of slide, you will see an important exam tip. Every gene occupies a fixed position on a chromosome. That position is called a locus. Different versions of the same gene at the same locus are called alleles. That's an important definition to remember for VCAA exams. Scientists usually represent alleles using letters. For example, capital A and a lowercase a. Notice something important. The capital and lowercase letters refer to the same gene. They simply represent different versions or alleles of that gene. For example, capital A might represent the allele for free earlobes. Lowercase might represent allele for attached earlobe. You will use this notation throughout VCE Biology, so it's important to become familiar with this now. Let's practice using allele symbols. Imagine the gene for plant height has two alleles. Capital T represents tall plants, lower t represents short plants. Since each organism inherits one allele from each parent, there are three possible combinations. Capital T, capital T, capital T, lowercase t, and lower t and lower t. Notice that capital T and lower t and the lowercase t and capital T represent the same genotype. By convention, we always write the capital T first. This notation becomes essential when we study genetic crosses. Some genes have more than just two alleles. A great example is the ABO blood group system. This gene has three alleles, capital A, capital B, and lowercase I. Even though there are three alleles within the population, each individual inherits only two, one from each parent. Pause the video and see if you can work out the possible alleles combination before we reveal the answer. Let's check the combinations. They are capital A, capital A, capital A, capital B, capital A, lowercase I, capital B, capital B, capital B, lowercase I, and lowercase I and lowercase I. Notice again that every person has two alleles. This idea is central to understanding inheritance. Let's finish with one final true or false question. An organism receives one allele for a gene from each parent. Think about everything you've learned today. Is this statement true or false? And the correct answer is true. Excellent. That's one of the most important ideas in genetics. One allele comes from mom and another one comes from dad. Together, those two alleles influence the traits that is expressed. We will explore exactly how that works and when we study dominant and recessive alleles in upcoming lessons. And now here's a final question. If a gene has the alleles capital B and lowercase b, which genotypes are possible? Think carefully. Remember that every person has two alleles. Pause the video before checking the answer. The correct answer is option D, with the capital B, capital B, capital B, lowercase b, and lowercase b, lowercase b. Notice that capital B on its own hasn't a complete genotype. Neither is lowercase b. A genotype always consists of two alleles because one comes from another parent. Understanding this concept will make Punnett square much easier in future lessons. Let's finish today's lesson with one final biology term, a locus. Locus is simply the fixed position of a gene on a chromosome. Imagine a street with numbered houses. Each gene has its own permanent address on the chromosome. That address is called the locus. Different alleles occupy the same locus on homologous chromosome. So remember, a gene is a section of DNA. An allele is a different version of that gene. A locus is the location where the gene is found on the chromosome. If you understand those three terms, you will find inheritance much easier. Congratulations on completing week 1 of VCE Biology Unit 2. Today you've learned about DNA, genes, alleles, chromosomes, genome, homologous chromosome, karyotypes, and loci, all of which form the foundation for the rest of the unit. Your next step is to complete the Almighty Choice Tutorial Student Workbook and Week 1 Homework Booklet to reinforce today's learning. In our lesson, we'll explore meiosis, discover why sperm and egg cells contain 23 chromosomes, learn how meiosis creates genetic variation. If you found this lesson helpful, please give the video a like, subscribe to Almighty Choice Tutorials, and share it with a friend who's studying VCE Biology. Thanks for watching, keep learning, and I'll see you in the next lesson.